Okay, like yesterday, remember we were having a scientific chat
Uh-huh.
and we were talking about network pharmacology.
Okay.
So definitely in network pharmacology, what we do is just we focus on the plants, right?
Mm.
And for each plant, you have what we call metabolites or active ingredients or active compounds, right?
Mm-hmm.
So for each plant, you have to extract those active ingredients, and the active ingredients are usually called they are called phytochemicals. I mentioned it. What we do is when you know the active ingredients, you now say, okay, which disease are you targeting?
Okay.
Okay, let's say you are targeting cancer,
Mm.
right? Or even HIV, like we mentioned yesterday, right? So what you do is you go to what we call a PubChem first. Yeah, first and foremost, you have to retrieve the target of your active compounds. Let's say you are working on moringa,
Okay.
right? So moringa, let's say they've extracted let's say about 30 or 40 or 50
Moringa.
phytochemicals, active compounds,
Okay.
right? So let's say if these phytochemicals are so many,
Mm-hmm.
right? You are not going to work with all those phytochemicals. So
Mm.
what you do screening first. Like yesterday I mentioned about the ATMET, right?
Yes.
Yes, you do the ATMET, which is the what? The final, the phytokinetic properties, right?
Okay.
So that's what we call Lipinski rule of five. So at the end of the day, if there is no violation to the Lipinski rule, so you
Mm.
can now select and use it to screen off some. Let's say you have about 20 or 30 which you can work on. So
Okay.
now, after you would have gotten that, you now go to PubChem,
Mm-hmm.
right? For each of the phytochemicals, you need to retrieve what we call SMICE. SMICE are just one-dimensional structure. We have 1D, we have 2D, and we have 3Ds, right?
Okay.
But now we're working with SMICE, which are one-dimensional structure of
Mm.
each of them. When you gather them, you put them, they form your the 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 phytochemic the plants, what you call the data set, right?
Okay.
Now you are targeting maybe HIV or you are targeting any other disease. I mentioned cancer, right?
Mm-hmm.
Okay, you also go to what we call a gene card to retrieve the genes in which that particular disease work on. And then in gene cards, the genes are already annotated. They're already put in there in the database. Let's
Mm.
say for cancer or for HIV, you might have up to 10,000 genes that they'll be working on. So now that forms another data set,
Okay.
right? So when you gather them, when you put them, and then when you gather that of the disease and that of the plants, right? So you will now have to clean your data set, right?
Mm.
After cleaning them, what you will now do is you find out the common genes among the plant
Mm-hmm.
and the disease.
Okay.
So that is when you now do your screening. We use Venn to have what we call the Venn diagram. The Venn diagram, now let's say you have 10,000 gene here, you
Mm.
have like 5,000 here. Now the common gene might just be like 200 or 300. So that's what you now do, and you select that, and that's you take for further analysis. We
Mm-hmm.
will end it here today, and then perhaps tomorrow we now do what we call the Venn diagram to see
Okay.
the common genes, and from the common genes you now get to get the hub genes.
Okay.
After the hub genes, you now start your further analysis. When you finish, then remember I mentioned the the biological functions, right? I talked about the gene ontology. I
Mm-hmm.
talked about the Keck pathway analysis.
Yes.
So when we finish all this, we also go to molecular docking
Yes.
to find out how the compound binds to the protein. So this will just take up in this series.
Okay.
Remember, we're just laying the foundation for this our series,
Yes.
and then yeah. So by tomorrow or next time we'll continue this. Thank you very much.
Thank you, sir.
It's all right. You're a good student and you're doing very well.
Thank you so much,
Thank
sir.
you. All right.